Hi, I'm Paul Campitelli, a first-year postdoc at ASU working in the Ozkan lab run by my advising professor, Banu Ozkan. Our work in the lab, and my work in particular, pertains to understanding the proteins in both the human body and other organisms at the level of single amino acids or even single atoms. We focus on the role mutations play on giving rise to disease, changes in bacterial resistance to specific antibiotics, or even identifying potential drug areas to inhibit activities of pathogens, such as COVID-19. The day-to-day activities of our work spans a multitude of disciplines, from mathematics to biology, chemistry, and physics, and activities from reading pertinent research, studying theory, setting up computational simulations, and conducting analysis. I'll be covering some of the broad concepts today in which we apply our computational tools to better understand the dynamics and thus the function of a large variety of proteins. DNA effectively represents the blueprints for any living system to carry out its biological functions, from the structure of cellular walls to the neurological activity to the construction, breakdown, and processing of small molecules. How does your DNA effectively carry out the instructions encoded into these blueprints? Well, largely, the answer is in proteins. Proteins are read off of the exome portion of a given genome, that is, the region which encodes for proteins. However, from one individual to another, this protein coding region of DNA can contain upwards of 10,000 non-synonymous nucleotide variants. Now, these are mutations which often result in proteins that, while generally performing the same function, may have some slight differences in biophysical behavior. That is, that my proteins and your proteins generally perform the same function, but may behave slightly differently, sometimes in ways which give rise to disease. Our efforts here are to understand how these small changes give rise to changes in protein dynamics. The subtle differences in dynamics must be understood in an effort to map out or predict how a DNA mutation may affect one individual over another. So what follows will be a discussion of some of the major computational techniques used to capture these dynamical differences through the use of simulation and computational analysis, which can be paired with experimental work to further understand our relationship between DNA mutations, protein variation, and protein function. Within current technological limits, experimentation provides the obvious advantage of physically assessing the structure and behavior of any given protein. However, there are limitations to experiment that have yet to be overcome by experimental technologies and techniques. Computational methods allow us to capture the behavior of proteins at time scales and length scales that may have otherwise been inaccessible in experimentation or simply prohibitively costly. In fact, oftentimes computational work can be pushed forward by mapping experimental data onto computational analysis, or even be used to inform experimentalists on how to proceed. For many computational biophysicists, myself included, at the heart of our work lies a theoretical approach known as molecular dynamics, simulations that allow one to capture the essential motions of a protein through computation by solving Newton's equations of motion while the proteins are subject to a variety of forces through self-interactions or interactions with their local environment, like water, ions, other proteins. This is described through molecular dynamics force fields. Through such simulations, we gain important insight into proteins subject to single or multiple point mutations. That is, say we have a protein of 200 amino acids in length, where we know certain single mutations, say one amino acid out of 200, can give rise to physiologically different proteins that may leave people at risk for various diseases. We can run simulations of all variants of a given protein and then subject them to further analysis. From molecular dynamics simulations, our group focuses on the dynamical behavior of the proteins by now applying an external perturbation through an analysis technique known as perturbation response scanning. Through this, we can understand the flexibility of proteins on the scale of individual amino acids, as well as how strongly coupled any two amino acids may be with respect to one another. One of the predominant types of protein systems we study are those associated with disease, and the relationship between amino acid mutations, dynamics, and the mechanics which give rise to diseased phenotypes. For example, we performed a dynamical analysis of human ferritin protein, in which certain mutations cause diseases associated with Alzheimer's and other neurodegenerative problems. Here we see that the flexibilities for disease variants, here shown in red, differ from the wild type in blue at functionally critical regions, while maintaining relatively similar flexibilities in neutral variants, shown in green. Note that these significant flexibility changes occur at regions far removed from the mutational sites themselves. This type of long-distance change in dynamics, which gives rise to critical changes at functionally important active sites, is a process known as allostery. In fact, with this human ferritin system, we find that the dynamic coupling of amino acids in disease-associated variants to an important loop is overall stronger, which disrupts the necessary allosteric regulations for this protein to function. This type of dynamical study can be extended to generic allosteric systems, proteins in which binding a molecule to one binding pocket can affect the behavior of a distant binding pocket, critical for enzymatic behavior. One such system is the PPIS protein, in which overproduction or underproduction can result in various cancers, or again, neurodegenerative diseases, respectively. This protein contains two binding sites, an enzymatically active site and an allosteric site, which enhances enzymatic activity of the active site. Here, we can also construct forced propagation pathways. When amino acid is perturbed, we find those close to it which respond strongly and in a similar direction. By repeating this process and identifying paths of connected amino acids, we can analyze the pathways unique leading from allosteric binding pocket to the active site binding when bound to an allosteric ligand versus the unbound state. In this manner, we begin to see unique signatures in which an allosteric pocket can communicate with the active site. And in fact, this type of communication direction has proven to be critical in allosteric regulation. The asymmetry in the coupling of amino acids, which bind directly to molecules in each pocket, also provides a unique fingerprint on the mechanics of allostery. In cases where proteins have evolved from an ancestor to a modern-day protein via several mutations, we can understand the respective physiological differences in a similar manner. For example, we've conducted studies on several families of proteins, such as GFP, green fluorescent protein, comparing ancestral proteins to their modern variants. In many instances, we find that the dynamics have changed in such a way leading to a phenomenon known as a hinge shift. So much like the joints in your skeleton can regulate critical movements of appendages, stiff regions of a protein, here shown in red, or hinges, have moved critically in ways which directly affect the protein dynamics and thus protein behavior. Now, for each protein shown here, the structure is conserved throughout evolution, but properties such as ligand binding promiscuity, stability and catalytic rates, and photochromatic activity can all change substantially as a result of a hinge shift. Seen here in GFP, the regions which were stiff in the modern ancestor, as compared to the modern extant, have changed fundamentally. We see that blue has shifted to red and red has shifted to blue. This effectively means that regions which were hinges have become more flexible, and regions that were more flexible have become hinges. While the structure is conserved, the protein as a result now will behave slightly differently than the ancestor. We've also applied this analysis to the SARS-CoV-2 coronavirus, compared to the SARS-CoV-1, to better understand the specific changes in key proteins of each system. In recent work, we have focused on a major potential drug target, the main protease. So current immunization methods target the spike protein, responsible for attaching viral cells to host cells. Upon infection, the virus produces a long chain of connected proteins called a polyprotein, which must be cleaved in specific sites for viral proliferation. The main protease is the protein which performs this cleaving. So when the main protease is inhibited, the polyprotein cannot be divided into functional units, and thus renders the virus biologically and biophysically inert within a host cell. In an article recently accepted for publication, we have actually identified key regions of the main protease which exhibit either a reduced or enhanced coupling to the critically important active site amino acids, here marked in yellow, in COVID-2 as compared to COVID-1. Similarly, some of these regions also overlap in terms of flexibility differences, and upon comparing the flexibility of the two systems when bound to known inhibitors, we have identified regions which may be used to bind additional drug targets. While immunizations currently exist, we understand that there continues to be enormous rates of COVID-19 infection, and subsequently, the search for inhibitory drugs remains necessary. And so with that, I'll go ahead and wrap up my talk. Here, I've provided a broad overview of some of the tools and types of work myself and my research group have conducted. As you can see, there is a huge array of problems which can be better understood through some of our computational techniques. The field of biophysics is vast, and oftentimes problems to study are limited only by the imaginations of the scientists working on these systems. And through the lens of protein dynamics, these problems, particularly important to wide-ranging study of human health, we can better understand and advance our treatment methods of a vast variety of physiological conditions. So I hope this brief talk has provided some insight into our work and our world, and hopefully some inspiration. I appreciate you taking the time to watch and listen, and please, as always, feel free to contact me if you have any additional questions or follow-up, and share or like the video if you feel so compelled. Thank you.